This week, you will inoculate several different types of media and perform a biochemical test. For most of these, you will need fresh colonies from an MSAT plate, so start one as soon as possible by taking an auto swab and spreading it out on an MSAT plate. If you have run out of MSAT plates, you may use a TSA plate instead. By the end of these activities, you will be able to explain the difference between selective and differential media and tests, explain the principle of 6 different media and 1 biochemical test that we use in this lab, inoculate a broth, inoculate an agar deep, and perform a catalase test. The purpose of these media and the test are to gather information about the bacteria, which may be used to identify the bacteria and teach you more about their biochemistry. Differential media are media on which most bacteria that we work with in the laboratory can grow. However, because of differences in the metabolism of the bacteria, different types of bacteria will appear different on the medium. Selective media, on the other hand, allow the growth of some bacteria while not allowing the growth of others. MSAT plates, for example, are selective because only benign bacteria from the oral cavity will grow well on this medium, while pathogens do not. You should gather the following materials for this activity. You will need a BAP plate. This is a bright red agar plate with black letters stamped on the back. A MAC plate, which is more burgundy in color and does not have letters stamped on the back because we prepared this medium ourselves in the Santa Fe College laboratory. PR glucose and PR mannitol are orange-red broths, liquid media in tubes, which should be labeled with the name. SIM and TGA are agar deeps. They have a small amount of agar to make them somewhat solid, while still allowing free movement of motile bacteria through the medium. The SIM will be more solid than the TGA. Avoid shaking these tubes. Lastly, the catalase test is a biochemical test and not a medium. You will need a glass slide and a few drops of commercially available hydrogen peroxide to perform this test. I will now discuss each of the different media, how to inoculate them, and how to read the results. As always, before you start, you should wash your hands and wipe down your bench. After you're done, wipe down your bench again and wash your hands. Once you've read the results of these media, you may discard the plates and tubes as described in the previous video. BAP is an abbreviation for blood agar plate, which is really just TSA medium with 5% sheep's blood added. Adding the sheep's blood makes this a differential medium that allows us to distinguish between the ability of the bacteria to break down hemoglobin. Hemoglobin is what gives the BAP the bright red color, so if the bacteria break down hemoglobin, the color of the medium will change. To inoculate BAP, select a colony from an MSAT plate and streak for isolation on the BAP plate. Incubate the plate for 1 or 2 days until the colonies show up, keeping track of the temperature. Some bacteria have enzymes that allow them to partially break down hemoglobin. The end products of the partial breakdown are green. These enzymes will be secreted from the bacteria into the medium, which causes the medium to turn green around the colonies. These bacteria are called alpha hemolytic, and the process is called alpha hemolysis. Other bacteria have enzymes to break down hemoglobin completely. In this case, the medium around the colonies on the BAP will lose its red color and clear up completely. These bacteria are called beta hemolytic. And lastly, gamma hemolytic bacteria do not have any of those enzymes and cannot break down hemoglobin at all. The red color of the medium will not change color. To read the results of the bacteria on BAP, hold the plate up to the light so that you can see the color better. If the medium turned green around the bacterial colonies, they are alpha hemolytic. If it cleared up completely, they are beta hemolytic. And if the medium remained red, they are gamma hemolytic. The next medium is Maconkey agar, or MAC. This is a burgundy-colored medium that has several ingredients that are of interest to us: bile salts, lactose, and neutral-red. These ingredients make Maconkey agar both selective and differential. The bile salts in Maconkey agar inhibit the growth of gram-positive bacteria while allowing the growth of gram-negative bacteria. This makes Maconkey agar selective for gram-negative bacteria. Lactose and neutral-red make Maconkey agar differential. Some bacteria can ferment lactose, producing acidic end products, which causes a decrease in the pH. And neutral red is a pH indicator that changes color at low pH. At high pH, neutral red is colorless, and at low pH it is pink. To inoculate Maconkey agar, you cannot use colonies from an MSAT plate because bacteria that grow on MSAT will be gram-positive, and gram-positive bacteria cannot grow on Maconkey agar. To see growth of gram-negative bacteria on Maconkey agar, you use the inoculation loop to collect bacteria from the faucet rim in the bathroom or kitchen or the sink drain of the kitchen sink. The bottom of the rubber disposal ring in the sink is a great source of bacteria. Then you streak for isolation on the MAC plate. Incubate the plate for 1 or 2 days until colonies show up, keeping track of the temperature. Any bacteria that grow on MAC will be gram-negative. If there is no growth, the bacteria you picked up were all gram-positive, or some of the gram-negative bacteria that cannot grow on MAC. They do exist. If there is growth, the bacteria are definitely gram-negative. From these gram-negative bacteria, we can tell whether they can ferment lactose or not by the color of the colonies. If the color of the colonies did not change, the pH did not decrease, which means that no acid was produced. These bacteria can therefore not ferment lactose. The result would be reported as gram-negative lactose negative or gram-negative lactose non-fermenters. If the colonies turned pink, the pH decreased, which means that acid was produced. These bacteria can ferment lactose. The result would be reported as gram-negative lactose positive or gram-negative lactose fermenters.